This is going to be talking about chapters 14 and 15, which is about genetics. And the kind of genetics here is the kind of genetics that people would call like breeding. So we think of animals and like dogs, and we think of plant breeding like agriculture. And then we also think of evolutionary change, talking about the understanding of changing that occurs during evolutionary timescales. And the type of genetics that is also called, it's also sometimes called Mendelian genetics, after a monk who lived about 150 years ago, Mendel, who studied peas and did breeding experiments in garden peas. It also would be called transmission genetics, because we're looking at the transmission of traits. And it's based on, it is focused on this inheritance that occurs from generation to generation. We can understand it from our perspective that we've gotten from chapters 17 and 18, where we looked at gene expression, and that is referred to as molecular genetics. And it's kind of like understanding inheritance from the ground up, from the bottom line. And so that's the modern perspective, and it's good to be solid on this modern perspective, because it makes this more traditional perspective much easier to understand. Genetics is complicated when you look from kind of the outside in, but less complicated when you look from the inside out. So we're going to start with there's a little more molecular genetics, kind of a molecular genetic review, where we'll be looking at each time we'll be talking about a gene. And we've got this idea that the gene is composed of two parts: the transcription unit, which, if it happens to be a gene that codes for a protein, has the coding region, and then the promoter. And the promoter regulates gene expression, and it regulates where, when, and how much of the gene product gets produced. When we say where, that means like brain or liver. When we say when, that means like embryo or adult. And how much means just the quantitative amount of the gene product produced. And when we look at genetics, a lot of genetics is based on the idea that you, typically in each of your cells, have a copy of a gene, given gene from your mom and from your dad. And so that you're often this combination of genes, and you might have one version from mom and one version from dad. And the versions of genes are called alleles, so we can define that. Allele is a version, one version of a gene, so it's a general term. And so you have two alleles of genes in all your cells, just about all your cells, because we've learned this as a diploid cell. And that's different than the sperm and egg, which are haploid and carry on just one copy. Now, an allele is one version. The versions can be different, and they can be different because of mutations. Now, when we think mutations, we always think that the mutations are bad, but it's mutations over evolutionary history that lead to different alleles in the population. So we have a population, like in the room, our classroom, we have a population. And we're all humans, so we all have the same genes, but we have different versions. And you and I are different because either we have different alleles or different environments that we grew up in. So that's this nature versus nurture kind of idea. nurture is the idea that we're different because we inherited some different alleles. And so genetics is all about that. Okay, so then the next thing is, let's talk about an allele. So one allele, and how could it be different? The answer, well, it could have some differences in the DNA sequence. So alleles are all about the DNA. They go on to, genes go on to make RNA and proteins, but those are based on ultimately the DNA sequence. That's what gets passed on between generations. DNA is the primary heritable molecule in all cells on Earth. And so one allele, its characteristics or its function is based on these four qualities. And one would be the coding region, so the product, and then 2, 3, and 4 would be regulation of the promoter, where, when, and how much. So when we say, we have two alleles, one from mom, one from dad, those two alleles could be similar or different. And if they differ, they would differ in those one or more of those qualities. Like that. There could be a difference in how much of the gene product is made. Okay? So there, and that's because the DNA sequences can be different in these locations and lead to these different quality changes. Okay? So then let's look at an example of alleles. And so we'll look at a red blood cell. And inside red blood cells, there are hemoglobin proteins. And the function of hemoglobin is to bind oxygen in the lungs and then release the oxygen into tissues like the muscles. So, in each red blood cell, there are, and we talked about this in class, there are tens of thousands of hemoglobin proteins. Where'd they come from? Your body made them. And in this red blood cell, in an immature red blood cell, and as it's developing the bone marrow, in the nucleus, there's a copy of the gene from mom and dad, and those get transcribed and by RNA polymerase and translated by ribosomes, and so these proteins represent some that were made from mom's copy and some that were made from dad's copy. Like that. So that you have these two discrete forms of the gene, and that's the genotype we defined in class. That's what you inherited, the plan. The genotype is the plan, and the phenotype is the product. Now, it's not the phenotype is, well, how well do you carry oxygen? So hemoglobin's function is immediately a little bit complicated, because we wouldn't just say that mom's hemoglobin binds oxygen better, like at a higher affinity, but that it has to be able to bind and release. So that gene products have often, a little bit of a complicated function. So it's a little bit hard to sometimes think about measuring the effectiveness, but we can imagine that in a population, we have some different sort of effectiveness of alleles. And so just theoretically, we could say, okay, so you can imagine that maybe mom's allele is functioning at 100%, and because of changes, maybe dad's allele functions either better or worse. Not just binding oxygen, but carrying it. So, again, a little bit complicated to measure, but down at that molecular measure level, we can imagine it. And in a population, there can be mutants at any level. It could be down around 10% or up around 115%. And so what you are is then sort of a blending or averaging, because you get a copy from mom and a copy from dad. You end up being a sort of a blending. So the genotype tends to be discrete separation, and then a phenotype tends to be a blending. So discrete genotypes in that you got a copy from your mom and one from your dad, and you'll send on to a child one or the other. We'll talk more about that. That's kind of the key thing of genetics when we get into it is, well, how do these two alleles work together? and they're kind of an averaging. So 100% and 10% results in output of like 55% on average. And so that's the phenotype at that molecular level. It's hard to measure level in some cases, and but we often look at other indirect phenotypes. And so let's just look at this idea for a minute. If we say that we had, sort of 100% activity and down from there, for hemoglobin, we just imagine what that's like. So as you go down in activity, that carrying of oxygen, there would be some things that would happen, right? So the person would first be healthy, and there's a general term for that scientifically when used in research that would be called wild type, just sort of the general quality character. And already in a population we're sort of hovering around this level. There'll be some organisms with a higher or lower percentage, but the idea is sort of that wild type is this healthy start. And then what if you knocked it down some? And you just imagine in hemoglobin that there would be some organs, like probably the brain, that would be very sensitive to oxygen, so that as you reduced it, there would be first these tissues that would be sensitive to oxygen, like maybe the person would get headaches because they weren't carrying enough oxygen to the brain. And so often that's the phenotype that's being studied. And that's another feature of genetics. It makes it a little bit complicated, is that we have the molecular phenotype, but it's typically more of a larger level phenotype, like the organism in its environment, that's being studied. So we could study how frequently do people get headaches, and we could correlate that with the hemoglobin alleles. And then, what's another one? Maybe it'd be a little less sensitive, but then it'd be true that there would be this thing with muscles. So there would be a kind of a fatigue, tiredness. And then to another level, there'd be some other organ, I don't know what it'd be, but maybe the kidney. So there would be some kidney failure, some failure in, removing wastes, like that. And it goes down, and eventually we get down to a level where, a person would be dead. And that death is a phenotype, lethal. Some genes would be alleles would be lethal. And so this is pretty common, and this is called pleiotropy. And pleiotropy is that one gene can affect multiple phenotypes. That, in this context, that's pretty common. Now, that's true for almost any gene, that it can have, as you knock it down in activity, can have multiple effects like that. And then another concept is that headaches are not caused only by hemoglobin. There could be multiple genes whose function is affected, whose function gives rise to an effect on phenotype. So lots of phenotypes are called polygenic, and that just means multiple genes affect this phenotype. And of course, the environment affects this phenotype as well, so it's a good one because there's often environmental factors too. But multiple genes affect one phenotype, and so that's complicating, but it's just the nature of this genetic study, is that it could be polygenic. Lots of polygenic traits are said to be quantitative traits. So just one more term, and we'll wrap this video up. Quantitative traits, just to introduce, would be a good example. A classic example of that would be height. So height is something that's affected by genes. That height tends to run in families. My parents were tall. I tend to be tall. My brother and sister tend to be tall. But its effects are due to they're polygenic. There are many genes that affect height. As a result, the trait tends to be quantitative in a population, in that when we see the phenotype, we see just a smooth distribution across the population from short to tall. So a smooth range, and that we can quantitate, we can measure the height of that. And it's a little hard to imagine, but these headaches are almost certainly also polygenic. They're quantitative, somehow if we could measure. And that's the thing. The next thing is that some phenotypic traits are easy to measure. Some are a little bit harder to measure, but yet they are also due to a gene basis. Okay, and so then all of these qualities are based on this up and down range of the nature of these alleles. And we have to look at these alleles in this pairwise combination. Well, we have this pair of alleles. How does it affect the phenotype? And they affect the phenotype on a case-by-case basis. So this allele impaired with this allele might give rise to headaches, but impaired with another allele, it might give rise to problems with not just headaches, but sort of more dramatic problem is the person can't get out of bed, and it's not because they're tired, it's because there's something wrong with their kidneys, like that. And so this has confused genetics, but it's really clear to see here at a molecular genetics level that the difference is this pairing of alleles, that different alleles cause these different effects. So there's a lot of focus now on the thinking about pairs of alleles, and so that's what much of this chapter is about, is understanding, analyzing pairs of alleles, how they get combined, and then what is the phenotype as a result. Good.